looks like we're ready to go. So the last thing I'll do is tell you about this. As part of starting this event, which we hope will be an annual one, we started a Inspiring Women Among Us award that is a non-academic award that women who face adversity in getting or returning to an education can apply for here at UNBC. So if you're interested in putting your extra pots of money that I know you all have, especially students, towards a good cause, pick up one of these and you can donate to the award. So sadly, I didn't get to do what I really wanted to do for this intro, but I'll try to do it for the next NRESi annual meeting, which is I like to go back through who gives these talks and check what our gender balance is, how many men and women we have as speakers, how many visible minorities, how many First Nations come up here in front of the room. And the reason I like to do that is not to make us feel bad or to point us out as somehow intentionally including a diversity of speakers, but rather to get us thinking about accidental exclusion in institutions. And what happens if we have a primarily, this is an example, right? So don't feel bad, primarily male faculty who taps into primarily male networks of scientists as we end up with a primarily male docket of speakers. So I wanted to bring Leigh here, Dr. Torres, as a counterexample. We went to graduate school together, so it's an example of what happens when you have a diversity of speakers. Who will they recruit? And we were lucky enough to do our PhD in an actually oddly female imbalanced, feminized campus because it was a marine conservation campus. So especially in the last 20 years or so, that's become a very female discipline with some exceptions. So that's another reason I wanted to bring Dr. Torres here, because the more technical aspects, like GIS, are still 50-50 or more male biased in terms of recruitment. But I would say generally, wouldn't you agree, the study of marine mammals is becoming more and more of a female pursuit? Even some of the very technical aspects like photo ID, which we heard about in today's Global Fridays talk. So that was one reason. The other reason is that we actually started an environmental group together when we were students, and this series of events is about recognizing inspiring women, but also seeking to inspire. And that's what we also tried to do when we started our environmental group and pushed for an environmental campus plan and ruffled a lot of feathers, but had fun doing it. So Leigh, we're really glad you can join us today from Oregon State University.
Thanks. Okay. I think I can turn this off. Can you hear me? Okay, great. Excellent. All right, well, thank you, Zoe, and thank you to UNBC for inviting me here. This is really, I feel quite honored to be here and to be part of this program. Zoe has always had this magnetism. She's managed to gravitate people around her causes and make people want to inspire each other, which I think is really special. So, yeah, it's great to be here. So, oh, that's not good. Okay. So I am a marine ecologist. I study the behavior and spatial ecology of predominantly marine mammals and seabirds. So I'm going to talk to you today about dots on a map. So, and that's just because oftentimes I'm just looking at dots on a map, GIS or in R or MATLAB, and it could really, the dots could represent anything. And so for many of you that work in terrestrial ecosystems, it could be any terrestrial animal. So this bluish figure here is what's called a stereograph, and in the 80s and 90s, they were the hit thing, and if you stared at it long enough in the right way, you could actually see an image in it. So if you stare at this long enough, there's a dolphin there. So good luck, go find it. But the point of this is that that's kind of how dots on a map are in spatial ecology, is that it can be a very complex effort to try and link behavior and animals and ecosystem to make sense of it all. And so that's sort of, I think, a lot of what I do. So, but I was asked before I jump into my science work to tell you about my evolution as a woman scientist. So I'm just going to take a brief hiatus to discuss how I got here. And so that's me on the right there as a young thing on one of my first soccer teams when I was growing up. And so I actually didn't realize how much of an influence my background as an athlete had on my career path until other people started pointing it out in me. But so it started off pretty innocently like that, but it turned into something more. So I really loved playing competitive sports. I can't really tell you why or whatever, but it was great in high school, and then I played Division I college soccer, and then even in grad school I took up ultimate frisbee where we went to nationals. So I've always really loved it, and I think it's always given me a sense of confidence and pride, and but I've also played and practiced and trained a lot with men and with other women as well that have confidence and egos, and it's a team effort a lot of the time. These are team sports for me, and so it was very important way for me to realize how to work with others, how to collaborate, how to deal with people's issues and biases, a lot of what we have to do in science today. So now I don't want to advocate that in order to be a scientist you have to play competitive sports. That's not my message at all, but I think it is really important to have outlets outside of your work, especially where you can gain confidence in what you do, and also where you receive constructive criticism and you learn how to take that in, because a lot of what science is about criticism. So whether you get a paper funded or whether you get funding or whether you get promotion and tenure, it's all about being basically criticized and taking feedback from your peers. So I think that's an important lesson to learn in whatever aspect of your life. And it can make you a better person, or better scientist and person, hopefully. So these are some words that I have been called throughout my life. And on a soccer field or an ultimate field, okay, I can take that. But I was often called these things and continue to be labeled these things off pitches, and for a long time I fought against it, because in my mind I wasn't doing anything different than what boys growing up would do, climbing trees, running, fighting back against things when I fought with my brother. And even today in my academic world, when I'm competing and doing what we scientists have to do, I'm still labeled these things, and I think it is because I'm a woman. A man doing the same thing might not be labeled these same things. Maybe they would, but I have tried lately, in the past 3 or 4 years, to embrace these characteristics about me that people see and not fight against, and realize, okay, well, this is part of me, and this is what is making me, successful in my career. So a few facts about women in science relative to men. So this came from a Nature article online. You can go here and plug in whatever position you want in fields. But I found this really enlightening. So this is sort of all a comparison of males and females over many years in all scientists and all positions. So you can see it's highly dominated by males. But I saw this and I was like, well, I work in sort of the life sciences. I would, think that it would be, a little bit less skewed. And maybe you all, as some of you here, young scientists, you look around your classrooms, you see there's probably maybe a better ratio of male to females. I know in my lab and around where we went to grad school, it was heavily dominated by females. So I changed these parameters down here, and I changed it to postdocs and then life sciences. And sure enough, the ratio is much more even, and that's great. And that was, what we would hope. But then I changed the position here to full-time faculty, and it switched back. And again, we have about twice as many males and females in full-time faculty positions. And so really this, to me, drives home the message of where we are with women in science. It's because of this female dropout rate. Okay, now there are heaps of stats out there about comparisons between males and females and salaries and so forth, but these to me drive it home, is that in the States, females receive half the number of PhDs, but only comprise 21% of full science professors. And there was a study that found that males and females without children leave science careers, research careers, at about the same rate, 20%. But for women who have kids or who want children, they leave research twice as much as men. And that, to me, is it. It's really at that postdoc period, that transition when women start trying to raise families, that balancing this career, a research career or academic career, really becomes quite demanding. So my recommendations, and I say from the trenches because this isn't necessarily my area of expertise. This is just, you know, I'm not a gender scientist, but I'm in there. I'm trying to do my research, but also deal with what I think of as subtle sexism these days. It's less blatant, I would say, than it was 10, 20, 30 years ago when women, braver than myself, you know, stood up and really rattled things, you know, when they were faced truly horrifying situations. But now I find things are much more subtle in how I deal with men and what I find how men deal with me in the workplace. So I have two recommendations. Is that one is that the postdoc to early career period is crucial, and we have to figure out how to focus on retaining women in the field as well as having families and raising families and caring for their families. So I think that that's a really an important period where, you know, funding or support or so forth needs to engage. And then the second point that I'll make before moving on to my sciences is that we shouldn't just talk about it, we should talk back. And what I mean by that is that having women in science groups and events like this is really awesome and it raises awareness. But I also think that we need to speak up, all of us, when we see subtle sexism occurring. And this is something I'm talking to myself here now too, because I was recently at a faculty meeting where I was having a nice discussion during a break with a colleague who was hired at exactly the same time I was, so a contemporary. And we were in the middle of this conversation, can't remember what we were talking about, but up walked this older gentleman, a emeritus faculty in our department, and he blew past me. He just butted into the conversation, started talking to my colleague, we'll call him John, and paid no attention to me. And I could tell that John was taken aback by this as well, and you know, he tried to bring me into the conversation, but basically ignored. I tried to interject, and you know, I was ignored again. And in the end, I just walked away. You know, after 5 minutes of trying, you know, truly feeling excluded, and you know, a bit of sexism going on. But looking back on that situation, I really wish that I had just talked back, that I had introduced myself, or better yet, that my colleague John had said, because I could tell he knew what was going on, that he had introduced me, you know, and brought me into the conversation, engaged me, and made this older gentleman recognize how his behavior was impacting things. So it's very subtle, I think, but there are lots of instances where we all see it, and we might not say anything because it might make the situation awkward or difficult later on. And I'm part of this too. So with that, I'll move on to some science. Okay, so I'm going to start by talking about spatial management of marine megafauna, which is essentially what I do. And I'm going to break it down here. So marine megafauna, because you all don't live on the coast here, just to tell you, is sort of all the whales, pinnipeds, polar bears, sea otters, sharks, turtles, seabirds. And, oops. Okay, so the question is, why do these animals need any specific conservation attention? And really, it comes down to one main fact, and then the derivatives thereof, that they're large. And before humans came along, that being large was a very good thing. They had evolved to be large so that they could avoid most predation, most types of predation, because once you get that big, you're hard to kill. But once humans came along, we actually targeted the larger of the animals because they were better effort ratios to reward and often gave us more product. But because they're large, they also have these other life history characteristics. So one, they're long-lived, and they have low natural adult mortality. They also have slow reproduction, okay? So they have low annual recruitment back into the population, and they have delayed maturity or age at first reproduction. So some sea turtles don't even reproduce till they're about 25, 30 years old, okay? And they also migrate long distances, okay? And they cross international borders, which makes management that much more difficult. And they also often aggregate to breed at predictable locations in time and space, which makes exploitation that much easier. Okay, so all of these things meant that they were easier to hunt, they were more targeted, and that their populations couldn't rebound or can't rebound that quickly from an exploitative event. Okay, so here's my definition of spatial ecology, Okay, it's a very simple one, but it's the investigation of distribution patterns and their drivers. So the first thing to know is that spatial, to me at least, doesn't just mean space, okay? Really, spatial ecology should be called spatio-temporal behavioral ecology. That's how I think of it. It's an ugly mouthful, so normally we all just say spatial ecology. But really, when we're thinking about animals and how they use their environment, we first think about space. So where is it? And this can obviously happen at many scales. So if we're thinking about a dolphin, you could say that its space is the Pacific Ocean, or you could say that its space is between the coast and the 100-meter isobath, and that's two different scales of saying where it is in space, where its habitat is spatially. And then we have time, so when something happens, and this is very critical, when animals migrate, when animals reproduce, when animals choose to feed diurnally. And then, of course, behavior. So animals use time and space differently based on their behavior. So whether they're hungry and feeding, or whether they're looking for a mate, or whether they're fleeing from predators or resting. So their use of time and space will be behaviorally mediated based on those needs. Okay, and then the last part of this sentence, spatial management of marine megafauna, is management. And this can take many different forms. Okay, but essentially here we need to integrate the spatial ecology of the animals and the human users to try and mitigate a lot of these space use conflicts, okay, that are happening more and more in marine ecosystems. Okay, and this can happen through protected areas, MPAs and reserves, as well as now we're trying to move more towards these dynamic time area closures that can happen at more finer scale. So instead of closing down fisheries across, a whole region, we can say, okay, well, let's just close it for these months or in this smaller area, which in hopes is to reduce burdens to, fishermen and so forth. So we're trying to find ways to make those effective as well as less regulatory burdensome. And then, of course, we can mitigate impacts, and by that is like putting TEDs, turtle excluder devices, on fishing trawls or torii lines or actual gear modifications to try and reduce bycatch events or any harmful activity. So when we're talking about management, we're really managing human behavior, obviously, and not animal behavior. And in order to do that in any effective manner, we have to have robust science. And then the other important element is enforcement, or some way to make sure that it's happened. So those are sort of the elements that I think make up spatial management of marine megafauna. So of course, oh, there we go. Of course, there's always the threat. So, I'm sure we all know of many threats out there in your ecosystem that you work in from anthropogenic impacts, but these are some of the major ones in marine ecosystems that impact marine megafauna. So fisheries, noise, ocean noise is on the rise, vessel strikes of large whales, habitat degradation and pollution from a whole host of sources, disease, invasive species, tourism, hunts, climate change, and then the cumulative impacts of all of these. And they're more, obviously, as well, but these are the ones that came to my mind as the most prominent ones. So in the next part of the talk, I'm going to discuss three case studies, three projects that I'm involved in that deal with these highlighted ones, these threats and marine megafauna. So the first one is going to deal with southern right whales and climate change. So southern right whales were heavily hunted, okay, and across the globe, but especially in the Southern Ocean, and so much so their populations were highly reduced. So they're hunted to near extinctions, and the populations, on a bright note, are recovering, and it's really great to see. And they're now definitely, we're seeing more and more calves. Their population extents are growing, but we really know very little about their offshore distributions. They are, when they aggregate to breed in coastal locations, and so that's great. We know where that is. We can protect that habitat, but very little is known about where they go the other 9 months of the year. And so, oops, sorry. Okay, so and then the other aspect is climate change, okay? So this is sort of the elephant in the room always. We know that things are changing. The ocean temperatures and patterns and ecosystems are going to be changing. But a lot of the questions are, how will these changes impact the habitat and distribution patterns of these predators? How will they, recognize change and be able to adapt to it? And can we predict and quantify these changes? So that was what this project was about, that we set out to do. So this is my study region for this area, so southern Australia here and New Zealand. And so like I said, we know they're breeding grounds. They're sort of along coastal New Zealand at these subantarctic islands here and along the south coast of Australia. But we wanted to know about their offshore distribution patterns. So we had to get some data, some distribution data. And this was it. These 48 locations. So not very much across this grand space. And you can see that they're quite clustered around their known breeding grounds anyway. So not a great data set at all. And that's because it's offshore. Not many people go there. There's no standardized surveys. You know, it's not that surprising. So I did a bit of hunting and thinking. And I said, oh, well, there was a lot of whaling, offshore whaling effort, okay? And so sure enough, it was big business. And because of it, the populations are quite reduced. So around New Zealand, we have about 2,000 animals, and around southern Australia, about 3,500. But the whalers were good businessmen, and they were all the captains were mandated to record their activities and where they didn't find whales. And they did this with their whaling log books. And so every day they recorded where they were, so their latitude and longitude, the date obviously, and whether or not they saw whales, as well as what species of whales. So this is a storehouse of amazing data. And these data come from 19th century Yankee whalers, so American whalers across the globe. And this great group here, the World Whaling History Project, they actually transcribed these, to me, illegible whaling logbooks. But they were great at it. And then I got to use the data, which was awesome because this is the data. And so it's heaps of data. So we have, and this is only 10% of the logs. They didn't go through it all, so just to give you a sense. So now we have about 25,000 records for daily locations and about 1,800 encounters with southern right whales. And basically, the bulk of the data is between, in the heyday of whaling. So if we just look at the presence locations across the study area and break it down by season. So we have spring, summer, and autumn. It's in the winter period that the animals are breeding. Okay, so we weren't really interested in where they're breeding. We know where their breeding habitat is. We want to know where they go offshore and where they're feeding. So you can see already just by looking at it that there does seem to be sort of this clump here, and then they sort of move south down here, and then they cluster here underneath the Chatham Rise. This is a bathymetric feature here called the Chatham Rise. So just by looking at the data, you could sort of see what's going on. And I'm just going to talk about this New Zealand region for now, just to keep the story somewhat simple. So I'm not going to get too much into detailed methods because I don't want to bore anybody. But for the people that are out there, this is what we did. We created species distribution models, which is a very common method used a lot in terrestrial ecosystems. In fact, most of these methods were developed in terrestrial ecosystems, and we're only sort of now applying them into the marine realm. But there's a whole heap of different kinds of species distribution models. But I used what's called boosted regression trees. And essentially what we do with the species distribution models is we try to relate where an animal is and is not found to its habitat or its environment. And that is basically done with something akin to this formula where the Y here is, for us, the presence and absence of southern right whales, which is our response data. And then we have a heap of environmental data, our predictor data. And we use a model, many different kinds of species distribution models, to try and understand these relationships between where animals are and the environment. So we did this using boosted regression trees, okay, which I think is a very powerful species distribution model, and it has two components, a boosted regression tree. The first one are the trees, the decision trees, where it tries to group the data, so your response data, into homogeneous groups based on your environmental data. So it has a tree that looks like this here. It breaks first at temperature and then at depth. But then what it does, it doesn't just make one model, it makes a whole mess of models, and that's the boosting part, okay? And at each next tree, it makes one tree, and then it takes the residuals, those observations that weren't fit well by the model, and makes a tree for that. And it does that over and over and over again, 1,000 or more times, and eventually amalgamates them all into one model that is more robust and has less error. So this is the method that I used. So these are the predictor variables, a totally overwhelming list, but many things listed here. I'll just briefly skip through them. So we have all these static ones, depth, slope, and then a range of depth and all these things. But then we have distance from certain geomorphology features, seamounts, valleys, lands. And these were our dynamic variables. So temperature, the mixed layer depth, so this is usually is associated with the thermocline, where above it is where productivity happens more, and it's a stratification layer in the ocean. And then distance from the subtropical front, which is a major current system in this region. So those were the things we threw at the model, but I'll focus on the things that came out as significant. And it basically came down to these 6 factors. So the mean temperature in the upper 200 meters, the mixed layer depth, the distance from the subtropical front, distance from ridges, depth, and the chlorophyll A concentration. So our models ended up having high predictive performance, we say, and mean external validation of 0.86, if anybody knows what that means. But basically, we made these models, and then we made prediction layers. And I'll show you better colorful pictures of this in a second. But the point here is that where it's darker is where the model predicted that there would be more whales. And then we used another data set of whaling data. So this is contemporary whaling data, the Townsend from also the 19th century. And then this JSV is the Japanese survey vessel. So this is modern sort of, they weren't whaling, they were surveying so that they could whale later. But they've gone around the world surveying for where whales are. And so that's what these stars and crosses are. And you can see that where the stars are tend to overlap with where the darker model predictions are. And so that's what we say, because that was independent data. It wasn't included in the model. So we say that the model is fairly well validated. So ecologically, we say that these oceanographic features and patterns are likely increasing the prey availability and density for the southern right whales. These southern right whales eat little copepods, zooplankton, and it's important that they forage in areas where the copepods are quite dense. Otherwise, it's not worth their time and energy, and they need these dense aggregations. So, okay, so another thing that often comes out of species distribution models are things that look like this, that are functional response curves. Now, I'll walk you through this, so don't freak out. So here we have the 3 different seasons, autumn, summer, and spring. And on the x-axis of each one of these graphs is an environmental variable. So here we have distance from ridge, temperature in the upper 200 meters, mean depth, chlorophyll A concentration, and the mixed layer depth. And then along the y-axis here, you can think of that as the probability of presence or the probability of absence. So if it's above the 0, it's that range of the environmental variable here, so we can look at distance from ridge, so is having a positive influence on whale presence. So here, this is a nice function here where clearly the whales are preferring to be closer to the ridge within about 500 kilometers and not beyond that range. So the take-home messages here were that mean temperature in the upper 200 meters between 10 and 13 degrees was good habitat for these whales in all 3 of our seasons. And then the mixed layer depth, they seem to prefer areas where the mixed layer depth was less than 40 meters, and that's because at that shallow depth, if the copepods aggregate right on top of that 40 meter thermocline, then they don't have to dive down as far. Imagine if that was at 100 or 150 meters, how much more energy they'd have to expend and how much more travel time to get to and from their prey source. So when the mixed layer depth is shallower, it's much more efficient for the animal. And then we found distance from these ridges, the Louisville Ridge and the Macquarie Ridge or the Chatham Rise was really important, and that's likely because of its function with currents in the region to aggregate prey. And then close to the subtropical front, especially here in this autumn season where there's this distinct peak right at the front. So we start to get a sense of how the environment is influencing the distribution of the southern right whales. So this is the prediction map. So we can use all these variables, put them together in the model, and make predictions of suitable habitat, okay? And so where we have the brighter red and orange colors, the model predicts good habitat as compared to the green. And the black line is just the location of the estimated location of the subtropical front in that season. So you can see that the model thinks the area along the Louisville Ridge here is quite important for southern right whales in the spring. The animals migrate south, pretty much surrounding the subtropical front in this region, and then they move west again, and aggregate just right south of the Chatham Rise, right next to the subtropical front. And then in the winter season, they go to their breeding grounds here at the Auckland Islands, or here, or along the coast of New Zealand. So basically, it looks like these animals make sort of a clockwise migration pattern around between their foraging period and their breeding period. So now we wanted to say, okay, now we bring in the climate change aspect. Okay, so now we have a better understanding of where they are now. But what's going to happen? Can we predict, or can we at least have any forethought about what's going to happen when climate change kicks in? So here we have the same current predictions that we were just looking at, but now we took out that layer of current temperature and added in a layer of the predicted ocean temperatures for the year 2100. Okay, and these are the predictions from that. So if we compare across seasons, we can see that this area here basically completely deteriorates, okay, and it's much further offshore. Okay, so you can imagine a whale leaving New Zealand or these subantarctic islands and trying to find foraging habitat and just not encountering it. Okay, and then in the summer, the area gets much more contracted, okay, smaller. And then, shockingly, in autumn, this area here that was so important is completely deteriorated, and it's not there. Okay, so the thing to keep in mind is that these are just predictions, and they're probably not that great predictions because they also don't include the changes in chlorophyll or the location of the subtropical front or the mixed layer depth, but because we're only changing one element. But you can see that how we can estimate that the ecosystem is going to change. And so the questions become, well, how are the animals going to find the habitat they need? Will they be able to adapt in time? Because what's happening, as we know, is that climate change is happening at a much grander scale than most animals have ever, rapid rate than most animals have ever, dealt with before. So will they be able to adapt in time? So just to wrap up this one case study, here we did the first description of southern right whale offshore habitat, which was great. And we also used historic data. Which for me was really exciting and proves that there's a lot of merit in trying to mine some of these old data sets, so in museum sources or old harvest records. But in terms of the conservation outputs, we described how ecosystem and fauna may respond to climate change. Again, this is not meant to be an accurate prediction, but an exercise to see how ecosystems are going to change and how a predator and prey might have to adapt to that. So we predicted significant change in the habitat, and we think that our results can be used to guide future research efforts. It's basically a warning sign for us to be cautious and aware of what may happen and how we have to be ready to adapt to that and adapt our management schemes. And to direct population monitoring of the animals and where they may and may not need habitat to be protected. And in general, just to aid conservation management of cumulative impacts of southern right whales, because they're also impacted by vessel strikes and entanglements and other issues. Okay, so that's southern right whales and climate change. You guys ready for more? So now we're going to talk about Maui dolphins and fisheries. Also in New Zealand, or in the New Zealand region, so you don't have to move very far. So the Maui dolphin is a cute little dolphin that's endemic to New Zealand, but it's highly critically endangered. There's an estimated only 55 adults left in this population, and that's mainly due to fisheries-related mortality in gill nets and trawling, as you can see here in this gruesome photo. And this has been going on for many years. New Zealand has an active gill net fishery, and they've put in lots of management effort and reduced the rates of this, but it still goes on, and every few years there is another mortality, which for such a long-lived species with low recruitment to the population is awful. Basically is driving this species to extinction. So what we wanted to know, well, okay, so I'll give you a bit more background. So, go back. Okay, so we're in New Zealand here. This is their population extent along the west coast of the North Island. They have a very coastal distribution, okay? And they're anecdotally correlated with people have said, oh, well, I see them a lot in turbid waters, near river mouths. But they said there had never been a quantitative study of their habitat use patterns or their distribution. So that's what we set off to do with these objectives, to describe the distribution patterns at a population scale, to identify the ecological relationships between their distribution and environmental variables, much like we just did with the southern right whales, and then to predict their distribution to new areas. And this is all with the goal in mind to try and help us figure out better ways and to conserve this species. So again, we're here on the west coast of the North Island. If we zoom in, there were 4 years of survey effort during summer months, and the black lines are the survey effort where the boat zoomed up and down, usually really close to shore, because that's where we knew where the animals were. The objectives of this study, the primary objectives, were to collect photo ID and tissue biopsy samples. So we get a little plug of skin that we use for genetic analysis. And then the red dots are where the animals were encountered. So you can clearly see there are clusters of where they are and where they're not found. So we wanted to know, well, what's in that habitat? Why are they there and not someplace else? Okay, so our first effort was to do kernel density estimates, which is a very common method of looking at an animal's general home range or distribution patterns. But kernel density estimates are often done with an equal x and y smoothing. Okay, so the equal smoothing on one axis as opposed to the other, so that if your sightings are these red points, you get these nice bubbles around it, all quite spherical or circular. So, but you can see here that most of the sightings are right up and along the coast. Okay, with different x and y extents, and our mean distance of a sighting from the coast was 0.8 kilometers, 80, not 80 kilometers, no. So what we tried to do was change the x and y. Okay, sorry. So when we used the typical method, this was the kernel density we got out, which was, you know, typical. You get this sort of fat balloon around most of your sightings. But we knew that this was wrong because this was insinuating that the extent from the shore went off about 3 or 4 kilometers, and we knew that that wasn't accurate. So we changed things. We changed our x and y smoothings, where we extended everything along the y-axis and contracted it along the x-axis so that the kernels turn out more oblong. And then the kernel ends up being like this. And so it's a subtle difference, but it ends up being really important, okay, because we know that these animals don't go offshore, so we didn't want to give the impression that that's what the data were predicting. So this is the customized XY smoothing and then the traditional. And then when we applied this across the 4 years, so these smoothing, this kernel density smoothings, we can see the black in each one of these is the kernel smoothing of the kernel density of the track lines, and then the colors are the actual sightings of, or the kernel density of the sightings of the Maui dolphin. And so you can see that in each year there's a cluster right here. This is the Manukau Harbor, and you can see that in each year the animals are found there. Okay, this is prime good habitat. Okay, and they're also in other years found in other places. And there is a slight trend, and it's hard to say anything conclusive because there's not enough data, that there is range contraction going on. You can see that it's quite, animals were found much broader extents in these years, and then this past year here. And that's when there was even survey effort. In this 2013, we had much less survey effort, so it's hard to compare that year. Again, not a great sample size, but you start to see a trend. Okay, so now we wanted to identify the ecological relationships between where Maui dolphins are found and the environment. So again, we had a set of environmental variables that we thought were relevant, as well as what was anecdotally reported. So we had distance to coast, depth, distance from watersheds, so that's rivers and harbors, sea surface temperature, and turbidity. Now turbidity ended up being a bit of a challenge because turbidity was only measured in 1 year, okay, during the 2015 survey. And we did this using a Secchi disk, which is one of these great low-tech data measurement tools that's been around for probably 100 years or so. And all it is you drop this disk into the water on a line, you will lower it down, until you can't see it. And then when you can't see that black and white, you stop there and you see how much rope you have out, and that's your depth of your turbidity. It's a very low-tech but very efficient and practical and consistent way to do it. So in 2015, the survey team conducted Secchi disk measurements, but that was only for 1 year. So what we did was we thought about chlorophyll, remotely sensed chlorophyll data. So remote sensing is an awesome tool. I'm sure you all use it in your terrestrial systems, and for marine studies, it's often very important as well because it's one of the only tools we have for measuring what's happening offshore. Now, people are often cautioned away from using chlorophyll layers in coastal environments because it's heavily skewed by sediment loads from runoff from rivers and dissolved organics. But in our sense, we thought, well, maybe that could actually work for us. Okay, maybe because it is, skewed by the turbidity in the water, we could use these images. So we related the Secchi dish measurements in 2015 to the log of the chlorophyll A concentration at that location from chlorophyll images. And sure enough, we got this nice linear relationship. And so we used this relationship in our models. So we used the log chlorophyll value from all years in our models. And that turned out to be quite effective. So I won't bore you with, we used booster regression trees again. I won't talk about the methods. I'll go right to the results. So sea surface temperature explained 30% in the model, and then turbidity, log chlorophyll, explained 21% in the model, while distance from major watersheds, 22%, and depth, 21%. Okay, and that, so these characteristics typify the habitat where the environmental variables that Maui dolphins are responding to most predominantly. And again, we have these functional response curves, and we can see that they prefer these warmer waters, higher turbidity areas, and then this really fine distance. I think this is about 10 to 15 kilometers from the mouth of a major watershed. Okay, so not right there at the mouth, but a little bit further away, and then it drops off rapidly. And then obviously shallow depth because they're right next to the coast. So we can stop for a minute and think about, well, what's happening here ecologically with these factors, particularly turbidity and distance from watersheds. And this is the study area along the west coast of the North Island of New Zealand. And you can see that, I hope you can see anyway, I know it's a bit hard, but these plumes, you can see the turbidity here within where my little cursor's going. Can you see that? Okay, yep, so more turbid water. And that little range is Maui dolphin habitat. So why are they there? And we had a good think about this. And this is what we thought about. They're predators. So it's not about prey all the time for these megafauna. It's often equally about predators. So their major predator out there is the great white shark. And there's a lot of them out there, as well as other sharks. And they have been seen dead on the beach with shark scars. They have been seen with shark scars in the wild. They've been the lucky ones that got away. And so while sharks are great predators and they frequently use smell and electromagnetism to find their prey, at short distances, they're visual predators. Okay? And so maybe the high turbid water offers them a evasive measure to avoid predation. But at the same time, the Maui dolphin, they need to feed, right? And this is their main prey, the red cod in the region. And they are found in not incredibly turbid water, but in coastal areas and reefs and so forth, just offshore. So our hypothesis is that these turbid waters offer a balance of predation risk, avoidance, and foraging opportunities, where maybe they're close enough to their foraging prey where they can just dart out and get it, or maybe they offer some protection in the turbid water where their prey are. We're not sure. This is a working hypothesis of the ecological function here. So then we move into our predictions. So there are two types of predictions when you're using, well, any model, but in a species distribution model. So you can have interpolation, which is where you use your model to basically make a prediction to where your data were from, your calibration data. So our data all came from this region, okay, within the black box here. And sure enough, it does a good job at predicting where we expect them. And that's what we expect. But the interesting thing is that these black lines here all are the management areas. So you can't trawl, you can't gill net, you can't do certain things all within here. And most of the habitat, these sort of bright areas, fall within that. And that's great, except for this one down here, which is actually a cause for concern, but that's another story. But so then we did an extrapolation, which is where we use our model to predict to areas outside of our study region. And that was around the whole North Island. And so when we did that, you can see these black areas. And mind you, there's a black area all underneath these yellow dots. And that was the black areas are our predicted good habitat around the North Island, the whole North Island for Maui dolphins. And the yellow dots are opportunistic sightings from public researchers, fishermen that reported seeing Maui dolphins since, I think some of these go back to like the 1820s. So they're historic data. But what we found was that 73% of these sightings occurred in habitat where we had predicted would be good Maui dolphin habitat. So it seems to be extrapolating well. And potentially this is information, because what we hope is that eventually the Maui dolphin will expand its range, you know, as the population recovers, and into some of these areas. Okay, and so what we need to think about is expanding some of those protective measures, the areas where they're not allowed to gillnet, into some of these regions. So hopefully this can help guide management efforts in the future. So just to wrap up this study, we confirmed the important habitat for this population, but it's also important to note this is only a summer model. Okay, so all models have limitations, and that's one of the main ones with this one. So we still don't understand, we don't have enough data to know what's going on in the other seasons. So we used 2 novel methods in this study, which to me is always exciting, you know, to face a challenge analytically and then to sort of overcome it. So we came up with this variable smoothing kernel density estimator, and then we used chlorophyll as a turbidity proxy. And then we identified suitable habitat for the animal to expand its range. And what our hope is that this information will be incorporated into habitat, into spatial planning management for the Maui dolphin. Okay, so my last example, which will be a somewhat brief one because this is definitely a project that is in motion. The other ones have sort of wrapped up, but this is somewhere at the beginning, I would guess. So this is an example of how sometimes things just fall in your lap, or not fall in your lap, but come out of nowhere, okay? And you're like, wow, that is really amazing, and a great research project, or it makes you move forward. So this is a blue whale in the great big blue ocean. And when I was living in New Zealand, somebody mentioned to me very casually that a seismic survey, a seismic survey is what the oil and gas industry does. They go out to sea and they pound the ocean floor with very loud noise, and it penetrates the sea floor, and that's how they find out where their oil reservoirs are. And it's a very common method of how the oil and gas industry finds their regions to exploit. So they had done a seismic survey out in this region in New Zealand called the South Taranaki Bight, and over 3 weeks they had spotted 11 blue whales. That is really unusual, okay? Like, that doesn't happen typically unless you're on a blue whale foraging ground or mating ground, although I don't know where that is, a mating ground. That's never been documented. But so that got me to start digging about what was going on here. And lo and behold, I ended up discovering a blue whale foraging ground. And I say in quotes they're discovering because I think that people actually knew it was there, especially those working on oil and gas rigs in this region. And they've all sort of come to me later and said, yeah, we knew that. Oh, yeah, we see blue whales there all the time. And clearly they knew it, but they didn't want to tell anybody. So, so the data I used, so this turned into a bit of a discovery, a sort of a mystery project for me, and I had to delve into all sorts of different data because, yeah, just to find out what's going on. So this is a bit confusing, but these black dots are data points that were reported to me of blue whale sightings. Oh, so first I should point out where the South Taranaki Bight is. It's between the South Island and the North Island of New Zealand, that area in there. So that's where we are here. Wellington is down there. So these black dots are opportunistic or seismic survey sightings of blue whales. These blue squares are oil rigs, so there's currently 7 oil and gas rigs active in the region. And then these red stars are blue whale strandings. So this is actually an unusual, again, amount of blue whale data in an area where the blue whale in New Zealand is currently recognized as a migrant by the New Zealand Threat Classification System, okay, which means that they don't think it exists in New Zealand except as it migrates back and forth between wherever and the Antarctic. Okay, so this is what I was up against. So blue whales don't occur here. Okay, so I was like, okay, well, there seems to be a lot of blue whale sightings and strandings in the area. But then I was like, well, what would they be doing here? And sure enough, there's an upwelling system, okay, that happens right here off the Kahurangi Point. And upwelling is where there's a lot of nutrient-rich deep water that comes up to the surface. It hits the euphotic zone, a lot of light, and turns into productive water. And then the currents push it up into the bight, and zooplankton blooms happen. And zooplankton is what blue whales eat. And sure enough, I looked into records from scientific surveys from the 70s and 80s that were zooplankton surveys in the region, and they found Nyctiphanes australis, which is a primary blue whale prey item in a foraging ground in Australia. So these were all clues that I was using to put my theory together that this was a blue whale foraging ground. And so then I looked at against satellite images, and here you can see an actual upwelling event, so that blue cold water, because it's coming from the deep, is cold. So that's that bit of cold there from a sea surface temperature image. And then a few days later, you can see a big productive bloom. from a chlorophyll image. And these black dots are, again, opportunistic blue whale sightings. So, and then this was my home run, I think, whaling data again. Okay, so here we have illegal Soviet whaling effort, okay, and what these is the blue whale catch, okay, and you can see that there's a peak of blue and purple right in this area, okay, relative to all around it where it's pretty low. And then the same with this Japanese scouting vessel data from a more recent period. Again, we have a peak right there. So I put, so this is an industrial map of the region. And so you can see, again, we have the oil rigs, okay? And then it is a major shipping route, okay? So Wellington's down here. Boats go off to the north. They go off to Australia or wherever. It's a big shipping route for New Zealand standards. And there's also a lot of fishing effort in the region. And I should say there's also a big seabed mining permit. So seabed mining is becoming a bigger issue in marine systems as resources are being extracted and used up in terrestrial landscapes. These companies are looking more and more into marine systems for their resources. And it's amazing the technology they are proposing to use because it hasn't been done yet, but it's right on the horizon. And New Zealand's first seabed mining permit was here. And actually my blue whale research had a hand in them being declined, which is a big thing. But of course, they're reapplying. So anyway, so this is sort of the industrial scene in the area. So I used all this data from other people, and I published a paper and that put forward this hypothesis that blue whales are feeding this area. And of course, it was met with lukewarm at best response from anybody in the industry. So I figured it was important to go out there and prove it. So I managed to cobble together some money, very little, and went out there for 5 days. And, oh, sorry, I have one more thing before I tell you about that. I forgot about this slide. This is a fun one. Okay, so this is, again, the industrial complex of the area. So again, we have the oil rigs, the blue dots. The black dots are whale sightings, and the red lines are areas of high vessel traffic. Okay, taken from AIS data, if you know what AIS is. Anyway, so what we have here is a blue whale. This is a very cartoonish example, but what I'm trying to get at here is the cumulative impacts that animals can face. We often study just one aspect of an impact, but I think more and more it's about the whole thing, what they're facing from so many different dimensions. So it's trying to find its krill here, Nyctiphones australis. So the two go, oh no. Oh, that is too bad. This is my, oh well, the two go round and round, you know, trying to find each other. And then there's ship traffic. You can't hear it, but there's loud ship traffic going on, right? Because vessels, these are massive vessels going through there, huge ocean noise, low frequency, right in the range of a blue whale. Okay, and then you have the seismic survey noise right on top of it, constant rhythmic pounding of the ocean floor, okay, and then you have this seabed mining going on, and then of course you have the fishermen, all in the same area where this blue whale is just trying to find its food, because if it doesn't find it, the blue whale has the highest prey demand of any animal, okay, they need to feed, okay, and there's only 4 documented blue whale foraging grounds in the southern hemisphere outside of the Antarctic, okay, so some go to the Antarctic, but some don't, and there's only 4. So New Zealand could be the 5th, but yet there's all this other stuff going on there. Sorry, that didn't work. That's a fun thing. Okay, so like I said, I managed to get some funding together, and we went out there for 5 days and found 50 blue whales. They were foraging half the time. We captured their prey. We looked at it on the echo sounder, and so we proved this hypothesis. But of course, it was only 5 days of data and really not that much except to make a lot of people raise their arms and shout. So there are lots of gaps, data gaps still. So we don't really know anything about the spatial temporal patterns of blue whales. Where exactly do they feed in the bight and when are they there? Are they there year-round or only seasonally or what? Residency and significance. So are the same animals there all the time? Do they move in and out? How significant is this foraging ground compared to other places? You know, is it that important? Do we need to protect it? And then who are these whales? What population are they from? And how are they connected to other populations in the region? So I've worked hard with colleagues to come up with a comprehensive research plan to try and address these questions. And I've tried to work with the various stakeholders involved, so industry, environmental managers, NGOs in New Zealand, the public, media, scientists, we're all stakeholders in this issue. And I've met with various forms of interest, various forms of ignoring me and pushing me back. Clearly some groups don't want me there. And I'm currently in this funding dance. I'm meant to go there. I have a boat hired for January 20th. I have a house hired. I have half the funding I need. Okay, the industry has declined to fund this work, even though we've had stakeholder meetings. I've tried to engage with them. Some of the NGOs have stepped up, but they don't have much money. So it's this interesting position that we're in. I was just having this conversation about how I'm a scientist, I'm supposed to be objective, but suddenly I'm in this role where I might be forced to play more of an advocate just to get the research done. So as I said, this is a work in progress, and I don't really know where it's going to go, but hopefully in good places. So this is my last slide that I'll wrap up with, which is the role of spatial ecology and marine conservation, but really you should just take that marine out, because this is the role of spatial ecology and conservation. So there's various applications of dots on the map, so I hope you got that out of this, and there's a lot more. I didn't even touch on movement ecology, so tracking data. But it applies to many different issues and conflicts, space use conflicts between where we are and what we're doing as humans and where the animals are and what they need. So it integrates lots of different disciplines, so genetics, behavior, population, connectivity, those sort of things. And it's useful at all scales. Spatial ecology, it's all about scale. And it's appropriate for most conflicts. And the last thing is that it has direct conservation outputs, deliverables to managers, maps. Maps are very interpretable. People like them. And that's what managers often need to point at things. And we need to protect this. You know, they go there. And maps are great for that. So I'll stop there and take any questions. Oh, right.
Well, thank you very much. That was a great presentation. In fact, I would have liked to have a whole hour on each of those species that you were dealing with.
Next time.
I see quite a few questions, but I saw Ken put his hand up early on.
Thanks. It was a very interesting talk. I was just curious how you deal with sort of a spatial autocorrelation problem. So if you know that the animals tend to be found in this particular area, then people go and they survey for them in that area. And then when you start looking at the correlation between where you find them and the variables, how do you deal with the fact that you're going to these areas with those particular variables, and so when they pop out in the models, how do you know that that's not just because you went to those places in the first place? How do you balance that out?
Yeah, I mean, you're exactly right. Spatial autocorrelation is a huge thing, and that's why ideally you include effort in your data analysis. So, well, in an ideal world, you do standardized surveys, line surveys across a marine environment where you're constantly surveying and you're recording absence data. But when that doesn't happen, you create pseudo-absences or background data. So basically, you know you kind of, the species habitat is across this region, you know, some hypothetical boundary, and then you can scatter points across that whole area, and you compare that to your presence data, and so you're saying, well, this is the available data, the background data, and this is where they were found. So that's one common approach to species distribution models, where you try and relate what we do know to what's available. And that sort of becomes what they're calling a presence availability model, as opposed to a presence absence model.
We walk the mic around the room so people at the end of the internet can hear the questions.
You have an amazing data set for the right whale. And if I understood that correctly, you had whaling data from the 1800s. Yep. And then you're comparing it to climate data, and that's from now.
Yeah.
And then that predicts where they're going to be. Yeah. Can you comment on that? Because that seems a little bit surprising. Yeah. I mean, that is a criticism we got from reviewers for sure, and it is something we talked about. So it's just based on the assumption that their habitat hasn't changed, or their habitat use patterns haven't changed since the 1800s till now. And because we wanted to include these dynamic variables. And so initially we thought when we did the proposal we would use these paleo records of environment. So there are corals and different ways that people have tried to measure the ocean and the characteristics of temperature and so forth, and frontal location from the 1800s. But those data records weren't available in our region. So we had to use these sort of modern modeled data. So
Again, I stress that it's probably not accurate. I mean, the predictions, but it's this exercise. I do think that the variables that we pulled out in those functional response curves are pretty reliable in terms of describing the animal's preferred habitat. And I don't think that currents have changed that much in their location between the 1800s and now. Now, what happens 100 years from now in 2100 is sort of anybody's guess, I think. So, but that's a good point. I mean, I can't argue, and all I know is that it's an assumption we made with the data.
If you can predict forward what climate is going to be like, can you back predict if what it would have been like?
No. I mean, those data sort of don't exist because people, people aren't making those sort of backward climate prediction models. People only are looking forward. So like that temperature layer from 2100 comes from the IPCC. It's not a layer that I made. And so when they have operation as business as usual sort of climate index, and then assuming industry, they make these layers that are available to scientists, but they only go forward, not backwards. But that would be interesting. Look at a less carbon emissions world 100 years ago.
It's still a really interesting case of basis stability.
Yeah, well, the oceans are typically stable, which is why things happening now becoming unstable are really interesting. Yeah.
In relation to sea surface temperature, would you be able to comment on effects of events like El Nino and La Nina on habitat use and pattern of behavior for right whales?
From what whale?
Right
whales.
Right whale.
Yeah. So those sort of cyclical things like El Nino or the northern or South Pacific oscillation, they happen every 7-10 years depending on which pattern you're talking about. And I think, so we sort of ignored that element here because we were thinking about a much bigger temporal scale. But clearly we do know that the ecosystem changes. Okay, so where temperature fronts are in one year with an El Nino is different in that region than where it is in a La Nina year. And so we do expect some change. So what we predicted was sort of an average distribution. What's interesting is that more and more now people are using El Ninos, like what's happening now off the West Coast with this warm blob. You all heard about the warm blob, okay. So, and an El Nino is here and is coming more, and there's a lot of weird things happening in the marine environment. Just in Oregon where I am, we had humpback whales. We still have humpback whales foraging right off the coast. We saw blue whales. I think animals are changing their patterns. They're looking for food because everything's messed up. We're finding tuna. We captured swordfish, 7 swordfish. I mean, this has just never been happening. All these sort of tropical temperate things. Oh, these temperate birds, these elegant terns from Hawaii showed up off Oregon. So there's a lot of weird things happening this year in the ecosystem that we're just observing, like, oh my, that's weird, never seen that before. Like the numbers of times I've heard that's never been documented before this past year, probably 20 times for different things. So really unusual, but what people are saying is this could be what climate change brings. Because if things warm, like a warm blob or something like this, these are the sort of changes that we might be seeing as animals searching more, going outside of what we perceive as normal to try and find their habitat, prey, mates, and so forth.
El Nino or La Nina type of event has been more predictable in the past, but as climate change progresses, then you can see more unusual cycles of El Nino or La Nina or coming back more frequently. So. That'll be another element, is more frequent cycles, quicker cycles. Yep.
So thanks very much for the chat. One of the questions I've got goes back to one of your first slides. So you noted that you are often characterized as ambitious and energetic and all those types of things. Fantastic,
I like that.
Fantastic catch at the ultimate play, by the way. I thought that was wicked.
Thank you.
I typically tackle people. I know I'm not supposed to.
That came after.
When you noted that that's how you're characterized, I guess as a male, an Alberta farm boy, I would take some of what you described yourself, or people describe you as, is amazing compliments, right? And I think that probably your role in sport and whatnot, and just that drive has done a really big thing here. And proud. I think this university's got some amazing female profs and stuff. But if you're characterized as that, as ambitious and energetic and whatnot, like to me, that's why you're successful as you are, because there's amazing energy and passion. Do you find that as a compliment, or do you find it as kind of a stereotype, or what are your thoughts on that actually?
Yeah, well, I think that was the point of me putting those words up, is that when I was younger, when people called me that, I was offended by it. And I think it was a part of the stigma I put on myself as a woman. I wasn't supposed to be termed that off the soccer pitch, and the fact that people still saw me that way I think I was offended. But I have come to embrace those words. Like, I do see it now as a compliment, and just like what you said, and I appreciate that. And I see that I use those characteristics in myself a lot more now that I'm more involved with science than I am with competitive sports. Yep.
Maybe one last question.
Oh.
A couple choices here. Two quick questions.
Hi there. Yeah, so you mentioned earlier that a part of your plan with Maui dolphins was to take tissue samples, and I'm wondering, did they get approved with such a critically endangered species?
Yeah. So it was a big permit process to go through, and there certainly was a lot of criticism about it. But in the end, certainly we did do it with permits, and that's actually how we have that estimate of 55 adults. Is that because unlike other dolphin species that are very well marked, we say they have nicks and notches in their dorsal fins, Maui's dolphins aren't very well marked, so we can't use traditional photo ID methods. So really that genetic recapture, because they went out and sampled in those sequential years, from that data is how we have that population estimate of just 55 adults.
Do I get to be last? Okay, wonderful talk, absolutely fascinating. I guess a historical question. As you know, I'm sure the science of marine mammalogy developed initially in close relationship to these extractive industries, whaling, sealing, and such. And now, of course, you're finding yourself, at least potentially with your research, often in contentious relationships with extractive industries, not necessarily whaling, but fishing and so on. Yeah. And even though sometimes they fund your work, at other times you really have to make the decision about when your work needs to go public, potentially, even if they don't want to fund it. How do you make that decision? I mean, because really the greatest, I guess, political leverage you have is the ability to go public with the sightings of blue whales and that data, even if they don't want to fund that. So how do you make that decision?
Yeah, it's hard, and that was that last thing I tried to touch on about the different hats scientists are having to wear lately is, I'm just a scientist, or I'm an advocate, or I'm going to go to the media. It's really hard. I guess for me, I really want my science to be robust, strong. I won't, with this blue whale thing, I was really hesitant to publish that paper because, because I knew people would argue against it and poke holes in it, especially the industry. But once I put it all together, I said, I believe in this. I really think it's true. I really think they're there. And so I think it's important that we take those risks and that we put it out there in the media and that we're not afraid to go up against big industry. And then when I went out and did the field work, that was even more gratification. When I first saw that first blue whale in the flesh, I was like, yes, they are here. So it's a very fine line I think we walk. I mean, I meet with industry often. I tell them about the work I'm doing. I've tried to be transparent with them. And, sometimes I think they're engaged, and sometimes I think it's just lip service. And then there's always the option of the media. They always want a good story, combating advocacy and industry, and so it's hard not to get carried away in that, but also funding is sometimes contingent about your conservation applications, so yeah, you always have to sort of play up one side versus another depending on your audience.
I'm really amazed to hear that with such an iconic species that funding isn't just flowing over you.
Me too. Do you know of any? Do you have any funding?
No, my flowing is pretty low.
Yeah, I know.
So I just want to thank Leigh Torres for coming here today. Thank all of you. I also want to point out that there are, or perhaps there were, one just left, a couple of male mentors in the audience brave enough to wear these t-shirts. So, be ambitious and courageous and brave and join them and buy one of these t-shirts and support our event and our award so we can continue bringing speakers like this to UMBC, we hope, year after year. The last thing I want to say is that as Leigh's host and friend, I'm going to commandeer her schedule and say we're going to the pub next. So if you'd like to talk to her about marine mammals or conservation or soccer, please join us. Yep. Thank you.
Come have a beer. Yeah.